Okay, so it's the screen for not reflecting what we have. Hello? Hello? Your pointers. Sometimes it takes long to. Oh, okay. Let me. Recording already? Yeah, it's recording. Yeah, I think once I build this, it automatically starts recording. Yeah. All right.
Very good. Thank you.
Thank you.
Hey, good morning.
But it's a festival, it's at the labor. Some year after some years, it'll take us a few.
Same with me.
Yeah, I know, but what's here? I just pounding it in. The word is.
Um,
Yeah. Oh, it's on though. It's not. Can you see?
hello. Can you hear?
Can you guys hear me?
Okay,
Oh,
good,
yeah,
good.
that's good.
All right, very good. Thanks. Um, Good morning, everyone. Um, Once again, my name is Diego Frenreich. I'm a cell biology and how about here? Better? I think it's a little low. How about now? All right. Thank you. I wish I knew how to fix things as quickly as you do. And today, I'm going to lecture about two interrelated topics. One is early stages of development, particularly pre-implantation development. And the second one is about stem cells, an emerging kind of topic over the past 10 years. Usually, and then you realize that to be a stem cell researcher, you have to know a lot about developmental biology, and you'll see that why I'm saying this. This is my email. You're welcome to contact me at any time. All right, and you can stop by my office as well. G667. Okay. So we'll start with development. We'll learn a few things about pre-implantation development, how we go from the egg or the fertilized egg to the blastula stage. Then we learn concepts about pre and post implantation development. Once the embryo gets implanted, we'll learn concepts about gastrulation, the formation of the third germ layer, the mesenchyme with the somites, notochord, concept of asymmetry. You know, we are very symmetrical on both sides, but not 100%. And we'll learn how this is programmed during development. And finally, we'll know a little bit about model organisms, why we study models, and we'll get to know the advantages and disadvantages of each of the models and the benefits, etc., etc. Okay. So this is schematic, and with the microscopy underneath of how an embryo, a unicellular oocyte, is fertilized, and it develops into a segregated form pre-implantation that is going to be ready for implantation. So we have a fertilized mouse egg that once it is fertilized, it triggers a process of cleavage in which one cell, one giant cell, this one, will divide through a mitotic procedure and will give rise to 2 cells. And then 2 cells to 4 cells, 8 cells. It's a rapid process, and 18 cells, 32 cells, and finally the blastocyst. The intermediate form here is the morula. And apparently all these cells are identical. They are called blastomeres. There are 2 features here that I want you to retain. One is, as you can see, this is at the right size of the egg, of the embryo, etc., etc. So we have a giant cell here, and we have 32 cells here, under or separated by. Let me see. I'm having trouble with this. That is contained in a layer of glycoproteins called the zona pellucida. This or this here in the schematic. Okay, this zona pellucida does not change throughout this early process. So how come you can have 16 cells or 32 cells in a place here that was designed to have only 1 cell? And can you guess why? What happens in between? Here, 1 giant cell, and then 16 or 32 cells containing the same volume. The cells cannot, we have an expanded number of cells here, but they are containing the same volume. There has to be something going on here. There's a process of compaction. Okay? The cells are not of the same size. This is giant. You can see it here. And this, as it is dividing, you will start seeing a process of compaction. And for example, you cannot distinguish here the cell borders, because there's and upregulation or expression of cadherins that will make tight junctions much stronger, so you can't really recognize the cell borders. And that's why 16 cells fit in a place for one giant cell, okay? So that's the process of compaction. And then this process of compaction triggers another process, which is segregation. Here, all the cells are identical, but here you can see morphologically that there are differences. So there are two main layers here. In development, everything is about layers, okay? So we have an external layer called trophectoderm, this one here in light blue, and then an internal layer, which is called inner cell mass. The inner cell mass of a blastocyst, made out of 32 cells, is the one that will give rise to the embryo proper. Just this, okay? Just this. And then we may wonder, why do we need this layer? And you'll see why we need the trophectoderm. Trophectoderm is really needed for implantation of EM. These are the cells that sort of invade the uterus, and then they implant into the uterus. So we have the inner cell mass of a blastocyst and the trophectoderm. This is just to put you in perspective. During human development, we have the ovary. We have a cyclic development of the oocyte. Every 28 days, there is ovulation here. So it goes from an immature follicle that matures, and then there is a rupture here, and the oocyte is being released into the fallopian tubes. Here, it is caught up by the fimbriae, sort of these digit-like structures catch the oocyte that is being released here. And this oocyte is just waiting for the cell to be fertilized by the sperm. Once it is fertilized, we start the process of mitosis or cleavage, okay? And during all this process of mitotic cleavage, you will see the zona pellucida. Zona pellucida, at some point, because of a pressure of all the cells that are compacted inside, it breaks up. And the egg emerges out of the zona pellucida at the blastocyst, normally or typically at the blastocyst stage, the last stage where the cells are segregated. What is one of the consequences of that? The embryo becomes sticky, very sticky, and then it can attach to the uterus wall that is waiting for the egg or for the embryo to implant, okay? All right. We'll move into this, and this is again to show you the process of cleavage. It consists of repeated mitotic divisions of the zygote, resulting in increased cell number. In human, cleavage is a slow 6 days process as the zygote moves through the uterine tube. Each cell of a divided zygote is called blastomere. All these cells, these and these, they're all identical or almost identical. And that's because it didn't go through segregation yet. If we were to make stem cells out of this stage, we would have, since these cells can give each individually, if I take one of the cells and I put it into another system, we can make a whole embryo out of it because these cells are all identical, right? That means that these cells, in theory, are totipotent, right? They can give rise to anything, including embryo and extraembryonic layers, like, for example, the placenta, the amnion. These are all layers that are extraembryonic that are extremely important for the development. But since, let me go further, since the stem cells are being made from the inner cell mass of a blastocyst, when the embryo is segregated, the stem cells are not totipotent, embryonic stem cells. They are going to be pluripotent, which is almost totipotent, okay? It's more than multipotent. It's more than 2, 3, 4, 10, and it's less than totipotent. These are the embryonic stem cells, the main feature of embryonic stem cells, pluripotency, okay? So I'm gonna move out of it. Morula, let me get to this part. This is a 32 cell stage compacted embryo, right? Once we start undergoing mitosis, the cells that are being created or that given rise of, they start compacting. Compaction also results in reorganization of the blastomeres. Compaction is a prerequisite to the next step, which is segregation, okay? It's all different concepts that I keep repeating. That's probably the best way to learn. Okay, so once we have, let me explain you a little bit more about the zona pellucida. The zona pellucida, at some point, because of the pressure of a blastocyst, because there is fluid that ingresses through the zona pellucida, the embryo needs to expand, and the zona sort of breaks up, and the embryo is sticky and implants into the uterus. Okay, I wanted to ask you if you can imagine what would happen if the zona pellucida is disrupted ahead of time, let's say at the morula stage. Let's go back, for example, here. The blastocyst disappear, the zona disappears here at the right time, and then the embryo without the zona pellucida can implant, okay, in the on the endometrium, yeah, a progestational stage. But if the zona would disappear here, the embryo would become sticky ahead of time and would implant ectopically, for example, at the fallopian tubes. And that would be a real problem in the on this particular pregnancy, okay? So the zona pellucida has to break at the right time when the embryo is in the uterus and the endometrium is waiting for the embryo to implant right there. Okay. All right, so then once the embryo starts the process of implantation without the zona, see, you don't see the zona pellucida here. You have the inner cell mass of a blastocyst here, and you have the amniotic cavity, a new cavity that is being formed here. The zona disappears, and we have the inner cell mass in light blue here that becomes epiblast, here, and then different layers, okay? So this process is being helped and organized by the trophectoderm. The trophectoderm gets here and starts the invasion of the blastocyst through the wall of the uterus, okay, the endometrium. And there's a mix of processes here that will end up in the development of the placenta. And this is a schematic to show you how the process of invasion takes place. So we have the trophectoderm that if you remember, I told you that it plays a crucial role in the invasion and of the embryo into the uterus, and the inner cell mass, which was going to give rise to the entire embryo, becomes a two-layered embryo. One, the epiblast here, which is made out of ectoderm, and one with the hypoblast here in yellow that is endoderm. So we have two layers. One is an endoderm and one is an ectoderm. There is a third layer that is missing here, which is which one? Mesoderm. Mesoderm, nothing about mesoderm yet, okay? So we have two layers. We have ectoderm in the form of epiblast, and we have hypoblast or endoderm, okay? Hypoblast is not definitive, but it doesn't matter. We have two layers here. So before implantation, we have two layers. The mesoderm, there's nothing about mesoderm yet. And there is invasion by the trophoblast here into the endometrium. Okay, the invasion is more pronounced. We have all these cell types called syncytial trophoblast, and then we have the cryotrophoblast, cells that start forming different types. So we have a trophoblast, and then that it becomes a syncytial trophoblast and a cytotrophoblast. These ones are gigantic cells that do not undergo mitosis anymore. But they play a crucial role in invading the tissue. Okay, and then we have these two layers, again, the epiblast and the hypoblast, and then new cavities. This is a cavity. Here is a blastocyst cavity that comes with the blastocyst formation. And this is a new cavity, the amniotic cavity. All these are layers and layers that are being formed through the process of implantation. Okay, so once the embryo is implanted in the mouse or in the human, and this new process that I'm going to tell you about can be also studied in the chick, and you'll see how. So essentially, the embryo is a ball of cells when it is pre-implanted, and it starts a process of segregation, but it's not symmetrical to any of the axes yet. So the development of the axis of symmetry is still needed at some point. Once the embryo implants, the mouse embryo, for example, or the human embryo implants, there is a new process which is called gastrulation. Gastrulation plays lots of different roles. One of those is to establish the symmetry axis, the anterior, posterior, dorsoventral, proximodistal. Okay, all these axes are being established during gastrulation. And also, there is the formation of a new layer, the one that was missing before, which is the mesoderm. Okay, the mesoderm, or the 3 germ layers, are being formed during gastrulation. So, axis of symmetry, and also the 3 germ layers during gastrulation. How does that occur? And it happens this way. So this is in the chick. Fortunately, we can study the chick for processes like gastrulation. Early events during development, can we study it in different models, different animal models. In the mouse, we can study gastrulation, but to study gastrulation in the mouse, we have to sacrifice the mother to open the uterus, right? So it became much easier to study the chick because that is external to the mother, okay? It's developing in the egg. So all we have to do is to cut the egg open. All right. So the process is through a streak called primitive streak. And this is the streak. So during that process, there is a morphogenetic movement of ectodermal cells from the epiblast. You remember the epiblast, derived from the inner cell mass. So everything is derived, concatenated. So I have inner cell mass of a blastocyst, epiblast, hypoblast, and now epiblast, there are cells that ingress through the streak and egress underneath the ectoderm, and they become mesodermal cells. They're mobile, they become mesodermal cells, the 3rd layer of the embryo. And this process will give rise to the mesoderm, okay? And the primitive streak ends up at the Hensen's node, and it goes like this. It starts at the caudal edge of the embryo, and it grows to the anterior part of the embryo, to the rostral part of the embryo, and then it comes back to the caudal part of the embryo. So the streak makes like a zipper kind of process. It goes from the caudal dorsally, and it goes all the way up and then all the way down again. In that process, there are several entities that are being formed right here, which is the neural plate that is part of the ectoderm that made that transition through the streak, and the notochord, which is right underneath the neural plate. I hope that it's very difficult to visualize all these processes. I hope you understand those. I selected sort of the best images or the best schematics to show you that. So during the process of a zipper of a primitive streak, there is the rise of a neural tube or neural plate, and then the notochord. The notochord is a structure like a rod type of a structure that is underneath the neural tube, and that exists only during development. So it's been formed here at gastrulation, and then it disappears. And it's a good thing that it disappears, because otherwise the embryo would contain pieces of a notochord and would form, that eventually would form teratomas, benign tumors. And you're gonna appreciate that pathology in a few fetuses that are born with some problems of notochord and teratomas at the caudal portion. So that is a big issue. But fortunately, in most of the cases, the notochord, in all of us, the notochord regresses or disappears. But when the notochord is there, it's a center, a signaling center, and produces lots of different things, particularly related to the formation of a neural tube and to the formation of the mesenchymal structures, like the somites. The somites are a group of balls. Let me just show you this. Okay, so before we go to the somites, I want you to get a good image of the zipper kind of situation. So we have a primitive streak that goes up, grows up here. Next to it or anteriorly from it, we have the neural plate, the notochordal process, then the streak regresses or zips down, and this grows a little bit more from the cells that that ingressed the primitive streak. And then eventually the primitive streak disappears, and we have the neural fold here. So all the cells, the ectodermal cells that went through the streak, some of those, they will form the neural structures, like the neural fold, the neural plate, with the roof, with the plate, etc., etc. And the notochord plays a crucial role in signaling and establishing all these structures. Okay, so this is another view in which you can appreciate the ectoderm here with the amnion here and the amniotic cavity here, a new structure. It will disappear in the fetus, but it will play a crucial role during development, the amnion. This is the ectoderm that will form the folds, the neural folds, the neural groove, and the neural tube. Okay, these are different instances of neural development. And this here is the notochord in a transverse view. Okay, this is like a rod that crosses the entire embryo. Okay, and this is a transverse kind of section here. This will emanate or create some signals that will allow the neural tube to mature, to form, and the somites to develop. Somites are the best evidence of the mesodermal structures, and they're grouped into like sequences of balls that are next to the neural plate. This is one ball here. Let me see if I can give you, yeah. For example, in this picture of an embryo, of a mouse embryo, you can, chick embryo, sorry. You could see all these balls here next to the notochord. This has been stained for the notochord, and next to them you have the sequences, a sequence of balls here. These are the somites, and this is probably the earliest evidence that we are, the mammals, including humans, we are segmented as well. We don't see ourselves segmented, but during development, we have evidence of segmentation. Then inside us, you can see the ribs, you can see the vertebrae, but not from the outside. So this is the first evidence that there is segmentation in a mammalian embryo, okay? We'll go back. Yeah, and the somites with different structures, sorry, that I don't want you to retain, but the somites will give rise to the dermomyotome, the dermis and the myotome, and also the ribs, the sclerotome. So these are all mesenchyme kind of structures that are derived from the from the somites. Okay, all right. this, you can appreciate that out of this will end up in, for example, 1 day later, for example, from the 20 somite stage, we end up in the 40 somite stage with most of the structures taking place. For example, in the cheek, the wing bud that will give rise to the wings, the leg bud that will give rise to the legs, okay? And the head, all the formations, etc. So, and we're not going to go further during development. I want you to learn up to gastrulation and the formation of the 3 layers, okay? All right, so something interesting. We know that we are segmented inside, but we are not perfectly symmetrical, right? We have the heart a little bit skewed to the left. We have different organs in different positions. How can this happen? That is established during gastrulation and during the process of the primitive streak and the Hansen's node that I showed you before. It turns out that within the Hansen's node and in other parts of the embryo as well, these cells have cilia. They are ciliated, okay? And they're not just cilia. They're cilia that they are all in synchrony. It's like the cells don't move, but it looks like the cilia wants to make them move. And what it does, the cilia starts rotating in an axis, all right? All of them at the same pace, but they are a little bit tilted. So they rotate a little bit tilted. What does this occasion in the embryo? What it does is it creates a gradient across the axis of symmetry from the left to the right, all right, on the fluid where the embryo is embedded in, okay? The cells rotate, they're a little bit tilted, that creates a gradient. And some of the factors migrate from the left to the right, and some of the factors migrate from the right to the left. And that's a simplistic but important way to understand how the axis of symmetry is somehow broken, okay? All right, so that is the concept of asymmetry. But remember that during gastrulation, what will end up forming is the axis of symmetry, okay? And the axes are made by rostral, caudal, dorsoventral, and proximo-distal, right? So these are the 3 axes of symmetry, and they happen during gastrulation. And in the mammal, that happens after implantation. Okay. Okay, so we have 10 more minutes. We're gonna finish a little earlier on this, but I guess earlier is better than late. We study model organisms during development. Why is that? Why do we study the mouse, the chick, and we don't study human development? Why? What is the main reason? It's obvious. Yeah, I mean, it's impossible to imagine that we'll study a human embryo, okay? So that's why we study the mouse, we study other species. So why do we study model organisms? They cannot be studied in humans. They have a similar set of developmental stages. For example, if you look at a mouse embryo, we have certain structures that are very similar to the human embryo. Okay, so it makes sense to study a mouse embryo. they share similar processes. As we went through, we have a similar fertilization process, a zygote formation, cleavage, morula, blastula, gastrula after implantation in the endometrium. Okay, we have 3 layers of development: ectoderm, mesoderm, endoderm, and we have similar underlying molecular mechanisms. The morphogens, the signal, the transcription factors, all those are being shared by the different model organisms. So this is a skeleton of a mouse embryo illustrating the vertebrate body plan. Okay? You can stain the ribs and the vertebrae and the different the ossification processes and all this in a mouse model. And here you can appreciate again the similarities between different models. For example, you have in a xenopus, what does it resemble to you? What stage of a pre-implantation embryo? One that we just learned about. After segregation. We have the blastula, right? We have the blastula here, right? With the blastocoelic cavity and the trophectoderm and the inner cell mass. We have a similar process here. So xenopus is a frog, okay? Frog and human are very different in the adult stage, but at the embryonic stage, it seems to be almost identical, okay? Very similar. So we can study the frog and make conclusions or generalized conclusions in humans. The chick, the process of gastrulation that I showed you about, that has been studied in the chick. Okay? So again, a chick and a human are a little bit different. Mouse development. You can see all the layers here doing gastrulation. Right? The 3 germ layers are here. And zebrafish, a fish. Okay? Look at these fetuses or embryos, very, very similar. And that's why we study all these animal models. Okay? And I'll give you like a list of features of each of the models, advantages, disadvantages, why we're studying these, which are the purposes. What I don't want you to learn or to pay attention to is the cycle. Don't look too much at the cycle. I mean, for curiosity purposes, if you want to look at them, go ahead. But this is not going to be in the exam. All right. What I want you to retain or to go over is the list of features of each of the models. For example, the amphibians, Xenopus levis or the frog. They grow in tap water. So that's so simple, right? We don't need to be sophisticated about growing a frog. Fertilized eggs can be easily obtained. In other words, we can inject the frogs with hormones to release the eggs, to facilitate the release of the eggs, okay? And to superovulate as well, okay? It could go 1.5x the number of eggs that can be provided. These are large eggs. We can look at them even without the help of a microscope, right? They cleave at 20-minute interval. The lower is the species that we study, the faster the cleavage or the cell divisions take place. They are resistant to infections after microsurgery. We can perform microsurgeries in the eggs, and they're resistant. We don't need antibiotics. We don't need anything. Okay. Useful for gene and protein expression and knockdown experiments, particularly RNA. RNA, everything has been done with RNA, or not everything, but most of the studies. And it's good for molecular and electrophysiological studies because there are not too many channels in the egg, and we can, for example, introduce an RNA that would produce a channel and study the quality or the features of that channel. That is one of the main uses of the amphibian egg. So that is for the frog. Then we have the birds or the chick. The chick is so versatile. We can study it for many features. Let me just go over this. Similar in morphological complexity to mammals. You can study gastrulation in the chick. Okay, even though there is no endometrium, there's no uterus, there's no implantation, these animals undergo gastrulation, and we can study the gastrulation process in the chick. They're easier to obtain and observe. For example, if we want to study mouse development, we have to sacrifice the mother to look at, or operate the mother to look at the embryo and study and follow up on the developmental processes of a mouse. But the chick, all we have to do is to put the egg in an incubator at the right temperature, and what we can do is to cut open like a window, manipulate the chick that is at gastrulation stage. For example, we can inject a beta-gal or a reporter gene into the notochord, into the somites. Then we inject that, and then we remove the pipette. We close the window with Scotch tape and put it back into the incubator. And the next day, we take it out again, we remove a Scotch tape, and we do the analysis that we want. So this is so simple and so cool. I find the chick a very, very versatile system to study fetal development. The other feature that is very attractive to me is in ovo electroporation. So how do we, so I mentioned, we inject the DNA into the somites, for example, right? How do we guarantee that the DNA will go into the cells and will incorporate into the genome? Well, we use in ovo electroporation. We apply electroporation with two electrodes, performing like an electric field there, and that would sort of electroporate in vivo the DNA into the cell. Okay, well, that's also very, very interesting to do. There are differences between the chick and the mouse, between the chick and the human. For example, as I told you, the chick embryo doesn't implant into the endometrium because there's no endometrium, right? There's no placenta in the chick, right? Like in a human or a mammal. The gases, for example, the oxygen sort of permeabilizes within the embryo. Okay, there's no placenta again, and they're exchanged by diffusion through the shell membrane, okay? The nutrients are obtained from the yolk sac, all right? We don't need the placenta, we don't need the maternal circulation. The nutrients are obtained from the yolks. So there are differences, but there are similarities. And depending on what you want to study, you can choose to study the chick or not. Yeah, you're not going to study, for example, placental development in the chick because there's no placenta, period, okay? All right. Going to move into the mouse. Everything has been sequenced in the mouse. And so we have plenty of genetic information. Not just the genetic information, but there are lots of knockouts, knock-ins, and genetic manipulations in the mouse. For example, these days, I want, for example, I'm interested in a certain gene. Then what I do is to look at the, for example, JAX labs are repositories of mice, and I see if a knockout is present there, and then I buy it. I purchase it. All I need is a grant to do that. And once I have that, then I can get access to any mouse model, knockouts, knock-ins, etc., substitutions, etc. So that's for the mouse. It has a short life cycle, 19 days. Generation of mutants by genetic manipulation, can do transgenics, knockouts, knock-ins, etc. It's closer to human than, for example, a chicken. All right. So if we want to use a more closely system, I would use the mouse, and then I could, if I want to get very close to human, I could use a large model, for example, a monkey or a dog or a cat, etc., etc. We can always step up in the complexity of the model before going into clinical trials, for example. And one of the setbacks is, of course, it develops inside the mother. Therefore, it's not easy to manipulation or visualization in uterus. Fish, that's another alternative. It has a short life cycle, and it's very well known. Well, first, the embryo is transparent. Use, for example, well plates to put one embryo in, or a certain number of embryos in one well, certain number of embryos in another well, etc., etc. And then we can apply a drug that will induce mutations. These are ENU. It's a toxic drug that will induce mutations. For example, in one well, it will induce mutations in certain kind of mutations. In another well, another kind of mutation. ENU stands for ethyl nitrosourea, and it will intercalate into the genome, into bases, and will produce those mutations. And then we can look at each of the wells and say, okay, which is the mutation that I'm interested in, okay? Or the embryos, the fish that I'm interested in. I'm interested in this one because it has a cardiovascular mutation, for example. And then I remove it from the well, I expand, and then I study the genome, and I look at where the mutation is, okay? And I discover a new gene, for example. The advantage, one of the advantages, is that the embryo is completely transparent. So I can, without manipulation, without staining, in vivo, we can look at the embryo and say, oh, this has an enhanced heart, for example, and then start studying this. So it's very versatile. Now, invertebrates, not only the vertebrates can be used as animal models, the invertebrates, the Drosophila originally was uh, studied like to completion, all the genetic information. And what is the main advantage of the Drosophila? The simplicity of the genome. For example, in the past, I studied the FGF factors. Okay, these are ligands, etc., etc. When I studied those, there were 10 FGFs. Now there might be like 30, and we'll keep sort of expanding on the numbers of isoforms. This complexity is a big issue when we study the mouse or mouse development. But in the Drosophila, there's one FGF. So you study one FGF, and you can study the whole signaling pathway with that one. So one or a few Hox genes, right? While in the mammal, if you want to study that in the mammal, you will have to sort of multiplicate this by 10 or 20 or something like that. So that's the main advantage of the Drosophila. And again, since this is invertebrate, the division, the cell division occurs very rapidly. So in one day, you can finish one study. Okay? And I think this is the last one, the nematode, C. elegans. It's just a worm. Okay? And one may ask, why is this important? What is the information I would get from the worm? For example, the IGF system, or the insulin-like growth factor pathway, or the whole signaling transduction pathway that is derived from IGF has been studied in part in the worm. And people observed that a worm who lives, let's say, one day, they started seeing worms that lived for 1.5 days, right? Between 1 and 2 days. And they said IGF-1 or IGF may be related or may be involved in aging, in the development, or it could sort of stop aging. And then they did more experiments, and they finally came up with those conclusions. Okay, so the worm is so important. The embryo is completely transparent as well, and it is very unique in a way that there are 500, look at this, 558 cells in a certain period of fetal stage or larval stage. And that is invariant lineage. So you can study, you can manipulate the cell number 325 and see what happens. Right? Everything is known about the embryo. And then you manipulate cell by cell. So I'm going to leave you with this. I think this is the last one. Yes. We're going to take a few minutes. We'll resume at 10:30. 3 minutes. Okay? 5 minutes. 10:32. Oh, any questions? Now or whenever you have them. Okay?
No.
What do we need to do?
Yeah,
Just
just
this?
expand it.
Okay.
No.
Okay, let's move on to the second lecture. This is about stem cells, and you will find during the lecture and at the end of the lecture that these two lectures are highly related, particularly to the pluripotent stage of the stem cells. So the main feature of the stem cells, two features. One is specialized cell that continually self-divide. That's one feature, and generate progeny cells for organ formation and maintenance. Generate progeny cells. So the stem cell has an extraordinary ability to do two things: to sense the environment and to say, okay, I'm going to generate more of these cells self-divide. But at some point, it says, oh, I need to, there is some wound, something that needs to be healed, and I'm going to generate more progenitor cells that would expand and finish the job. So these two features characterize or are common to every stem cell that we have in an organism or outside the organism, like, for example, the embryonic stem cells. They are found in different tissues at all stages of life, before and after birth. And that's important because people tend, not now, but 10 years ago, when the boom of stem cells, people thought that all stem cells were identical or similar, so you can treat with mesenchymal stem cells, and if you don't have them, so you use hematopoietic stem cells. If not, you use iPS cells, etc. That's not true. The cells vary in their properties depending on where they have been born, okay, or they are located. So the stages of life, before and after birth, there are different types depending on the location of the body. A mesenchymal stem cell will not be prone to make hematopoietic cell, and a hematopoietic cell will not be prone to make cartilage, for example. The main classification, not the only classification, but the main classification is embryonic stem cells, which are obtained from very early embryos, like blastocysts. Now you are experts on blastocysts. And adult stem cells observed within tissues of children and adults. We all have adult stem cells in different places. For example, in our bone marrow, we have hematopoietic stem cells. They are acting through the entire lifetime, okay, with different, they age also, they have different properties, but hematopoietic stem cells, the neural stem cells, responsible for expansion upon a certain disruption of a tissue, a contusion, for example, concussion. We have even cardiac stem cells that is very controversial, but half of the cardiac community think that there are stem cells in the heart, etc. So we have embryonic stem cells and adult stem cells. And these are the objectives of the today's talk on stem cells. We are going to go through the embryonic stem cells first, and with emphasis in how do we obtain these cells from an embryo, okay? And what are the consequences of sacrificing an embryo? Nuclear transfer embryonic stem cells. These are embryonic stem cells, but they match the immunological profile of the individual. So they are patient-specific embryonic stem cells. All right, very, very important. Then we have adult stem cells with the whole variety of adult stem cells. We have induced pluripotent stem cells, that a wonderful biological concept for which the scientists obtain the Nobel Prize. And I'll talk about this a little bit later, but to replace embryonic stem cells or to provide a new alternative to the embryonic stem cells. Then transdifferentiation, a new concept or a 10-year-old concept that hasn't panned out very well lately, but as a concept, it's very powerful, and I want to bring it up. Then we have a new type of, it's not a stem cell, but it's something that we can do with the stem cell, can make a synthetic embryo. We can, it's like It sounds weird, but it's an embryo that it's been formed without fertilization and without an uterus. It doesn't implant. It's being grown in a bioreactor. And I'll give you a little bit of that. It's a breakthrough, and we should pay attention to that because we can study that embryo without any or with much less ethical concerns, because that's not an embryo. It's an embryo like. Well, you see all that. And then we have similarities and differences between the different types of cells, advantages and disadvantages. Not every cell is identical. All right, let's start with embryonic stem cells. They are culture cell lines derived from the inner cell mass of a blastocyst. You remember, the blastocyst, the last stage of the pre-implantation embryo, segregated two layers, inner cell mass and trophectoderm, okay? If we remove the inner cell mass of a blastocyst and put it in culture in very, very rich conditions, these cells can give rise to the embryonic stem cells, all right? And since this stage, this embryonic stage is pluripotent, we can have embryonic stem cells that are pluripotent, okay? What does that mean, pluripotent here? It means that we can make potentially every cell type of an embryo or of an individual. But there are cells that can't be made out of embryonic stem cells. Can you think of a cell that cannot be made? Looking at the image here, this embryo is segregated into two layers. The inner cell mass, and we make the embryonic stem cells out of the inner cell mass, and the trophectoderm. Which cells cannot be made with embryonic stem cells? Just guess. Yeah, the placenta, the extraembryonic tissues, because there is a segregation already. If we wanted to make placental stem cells, which cells, which layers should we use? This one, the trophectoderm, because they are responsible for making the placenta. And that's how we're thinking, and that's why the developmental biologists and the stem cell researchers have to come together to study, or we have to know developmental biology to be a good stem cell researcher. All the extraembryonic tissues, for example, the amnion, the chorioallantoid placenta, all those tissues cannot be made with embryonic stem cells that are derived from the inner cell mass of the blastocyst. And that's why these stem cells are pluripotent, as opposed to totipotent. They are not totipotent. They cannot make placenta. Okay? All right. So they can be grown indefinitely in their undifferentiated state. That's a wonderful, very powerful concept. I can derive embryonic stem cells, and I can have them growing and growing and dividing on the dish forever. I can also store the cells in the -80 or in a cryopreserver. 10 years later, I can take them out, and they will still be growing in the dish forever. That's something that we cannot obtain with adult stem cells. Only with embryonic stem cells. We can grow them indefinitely. And the other feature that I just told you is that we can make every cell type of an organism. Meaning that if I make an embryonic stem cell and and check how good they are, they potentially they can make any cell type of the embryo, okay, of the organism, but not the placenta or the extraembryonic layers. Remember that. Embryonic stem cells, pluripotency. All right, they are capable of differentiating into all cells of the adult body, pluripotency. The source of embryonic stem cells is the inner cell mass of the blastocyst, okay? The ESLs can only be derived from cells found in the embryo. When we talk about mouse ESLs, that's nobody cares about whether to use an embryo, a blastocyst, for embryonic stem cell purposes. But when we talk in human experiments or human research, the problem is that if you want to make human embryonic stem cells, you have to sacrifice an embryo. And that's a huge ethical concern. Okay? It can be done if the donors of the eggs or donors of the embryo consent, and there are proper documentation for that. Okay, it can be done. In the past, it could be done only without federal funding, but now you could do it with federal and private funding, but you have to have all the documents in place. Okay, we can apply to grants using embryonic stem cells. That's not a problem, but we have to report that. We have to check one box and say, yes, these are the cells that I'm using. Okay, it has to be. People are very, very particular with embryonic stem cells, but it can be done lately. Okay, so and what's the ethical concern? That potentially that blastocyst that we are using, if you are putting back into an uterus of a surrogate mother, can develop a life. All right, and that's the problem. Again, as I told you before, the ESLs remain the only stem cell type demonstrated to produce all the approximately 200 plus cell types found within the adult body. But again, if I asked you, for example, can you make amnion out of embryonic stem cells derived from the inner cell mass? What's the answer? No. Okay? Yeah, that's the answer. So you can make pancreatic islet cells, for example, beta cells to produce insulin, something that it's not very doable with adult stem cells. So there are advantages of using embryonic stem cells. You can make hematopoietic cells, cardiac myocytes, neuron, hepatocytes. So you can make cells from the 3 gem layers: ectoderm, mesoderm, endoderm. Okay, and it makes sense because these cells were obtained from the inner cell mass before the segregation of the different layers. So there's a lot of embryology here. Okay, human embryonic stem cells and IVF, the in vitro fertilization clinic, very interrelated. Not all the eggs will fertilize and develop properly into blastocysts during an IVF procedure, okay? Or the couple use, for example, 3 eggs and put in the cryopreserver 3 or 4 or 5 different eggs. And at the end, they want to discard these eggs, and they're not going to use them for for example, for another pregnancy, and therefore they are used to create, for example, the different cell types. So not implanted blastocysts are frozen for another pregnancy and ultimately discarded. These discarded blastocysts have been used to derive human embryonic stem cells in the laboratory after approval of the donors and oversight by the appropriate IRB or ethics committees. And this is essentially a schematic of making embryonic stem cells utilizing in vitro fertilization. Here, we have the egg in an IVF clinic. We use the sperm, there is fertilization, and then make the ESLs upon removal of the inner cell mass, and then differentiate into any type of tissue that is needed and put it into a patient. Major features of the ESCs or ESLs. They are pluripotent, potential to differentiation into all functional cell types of an embryo. For example, the ESLs can make into a neuron, into a cardiac myocyte, a muscle cell, a blood cell, blood vessel, endothelium, for example, skin fibroblasts, you name it, okay? Different types of cells. However, and that's something that I haven't mentioned yet, they have potential for teratoma formation. A teratoma is a benign tumor. It's not a tumor that that is malign, okay? But yet it is benign, but nobody wants, for example, to use cells to treat a condition and start creating, for example, in the heart, cells that will form a complete different pattern. So that's a problem of embryonic stem cells. And in general, it's a problem of every pluripotent stem cell. In this particular situation, the embryonic stem cells, but we'll see that there are iPS cells, for example, that they're reprogrammed and they go to a pluripotent stage again, and pluripotent stage is associated with teratomas. Okay? So every time you hear about pluripotency, that equals teratoma potential. That's important for you to retain. So research with embryonic stem cells will help to discover the factors necessary for regeneration and repair of the tissues. So we could use the embryonic stem cells not just to inject into a mammal, for example, but we can do drug discovery. We could do many different things. We can grow entire hearts. Let's say we have embryonic stem cells and we use them with cocktail A to make cardiac myocytes, cocktail B to make endothelial cells, and we keep growing on the cocktails and we can produce 3D structures. And that has been done with embryonic stem cells. For example, we grow kidneys, livers. A few days ago, I read about making trachea, but I don't remember. I think it was made out of embryonic stem cells. So this is an emerging kind of, an evolving kind of process or field, very powerful. We can make polymer matrices. We can use 3D printing. We can use the Kensai system that we used when a few years ago in the core, in the stem cell core. This system has, for example, a big needle that catches, for example, a ball of embryonic stem cell derived cardiac myocytes, right? So there's one column of cardiac myocytes, and then another needle that catches another ball of cardiac myocytes. And then you put one column, two columns, three columns, four columns, and then rows of cardiac myocytes. And then you pepper with endothelial cells and fibroblasts, and you given the topology that you desire, and then you can use those cells or those structures to patch, for example, a heart of a newborn baby or a fetus that has, for example, the Swiss cheese syndrome, which means that it has holes in the septum. So you can use those to patch if you are using the right kind of cell. You can make iPS cells from that fetus, patient-specific, or nuclear transfer embryonic stem cells from that fetus and catch that those septi with its own cells. There is a lot of potential. And we have to team up with bioengineers to see what the topology will be, et cetera, et cetera. All right, let's move now into the adult stem cells briefly, and then come back to the embryonic stem cells, okay? So we covered embryonic stem cells. They are pluripotent. We can do lots of things. Adult stem cells are also very good, and depending on what we want to use them for. For example, if we want to make hematopoietic cells, why not use hematopoietic stem cells to derive specifically the cell type that we want? Right? Because if we use embryonic stem cells, for example, we can make hematopoietic stem cells or hematopoietic cells, but we can make many other things, and those will contaminate the tissue that we are making, and that's not good, okay? Depending, if you're using for therapy, maybe the best thing to do is to use hematopoietic stem cells to make hematopoietic cells. Okay, so they are derived, the adult stem cells, they are derived from different parts of the body: brain, bone marrow, fat, for example, adipose stem cells, neural stem cells, and we keep discovering new source of stem cells. They have different properties, as I mentioned. They can be multipotent, tripotent, bipotent, but never pluripotent, okay? This pluripotent feature is very, very specific or ascribed to the embryonic stem cell. So the most popular ones are the hematopoietic stem cells as adult cells. These are adult stem cells found mainly in the bone marrow of adults. They form all the blood cells, red and white blood cells. We can make lymphocytes, for example, with the hematopoietic stem cells. They are required for the daily blood turnover for life, okay? And for fighting infections. And again, they have to create progeny, but they have to self-divide when time comes, when there is a depletion of the pool of stem cells. So they have to sense the environment and make the appropriate kind of cell. They're easy to obtain. They are bone marrow, either by bone marrow aspiration or stimulation with cytokines or cytokines like GCSF, granulocyte colony stimulating factor to move them to peripheral bloodstream. So we have them in the bone marrow. We can either resort to biopsies or just inject certain factors that will release those hematopoietic stem cells into the circulation, which is very simple. Then we have to draw some blood and then separate. Hematopoietic stem cells were the first stem cells to be used successfully in therapies for blood cancers, leukemia, and other blood disorders, even without knowing certain clinics, hospitals, they use cells without knowing that there were stem cells there, right? Hematopoietic cells. The embryonic stem cell concept, like 50 years ago, 100 years ago, it was not known, right? And people were using hematopoietic stem cells to treat diseases, leukemias, etc. So they are very, very potent, but not potent in the term of pluripotency. They are multipotent, but they have a lot of potential. Okay. This is a typical tree of an adult stem cell kind of schematic. We don't have, as embryonic stem cells, the property of making anything from the embryo. You don't see pluripotency here. You see multipotency. You can make 5 different lineages, 10 different lineages. Maximum, here you can have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 15 maximum. But it's very important too, right? And these cells, these hematopoietic stem cells, they are very mature and they can resemble the maturation status of a patient. While the embryonic stem cells, one of the main problems is that they are immature. The fact that they're immature will give you advantages and disadvantages, both, but they're immature because they are being obtained from embryonic stem cells, right? So if you make, for example, a cardiac myocyte out of embryonic stem cells, they will have, for example, sarcomeres, but they would be a little bit immature. And if you do any transplantation experiment in the mouse, or these cells, if you make, for example, beating cardiac myocytes in the dish, you will have them beating at a much faster rate than an adult cardiac myocyte. Instead of being 60 beats per minute, they could beat at 120 beats per minute because they're immature, because the fetus beats at 120 beats per minute. Okay, they are more like fetal kind of stem cells. All right, how do we treat a leukemia patient with an hematopoietic stem cell? Well, instead of making embryonic stem cells, you go to the tissue, the bone marrow, extract the cells that you are interested in. Here you have two different kind of cells. You have the hematopoietic stem cells. Here, these round kind of cells that they don't attach to anywhere because they are cells designed to be traveling the vasculature, so they don't like to attach. These are the hematopoietic stem cells. And the other ones, the stroma stem cells that will give rise to the mesenchymal stem cells. So you have a mix of cells and different variants. So you put them in culture, and the hematopoietic stem cells, while the stroma cells will attach to the bottom of the plate, hematopoietic stem cells will not attach, will never attach, right? So then you take the supernatant and you purify them a little further using cell sort techniques, and then you get the type of hematopoietic stem cell that you want. And then you can use them. For example, you can inject or transplant into a patient that was removed from his own batch of cells because he had leukemia, for example. And these cells, these hematopoietic stem cells, have the extraordinary ability to recognize the place and home into the bone marrow. So they go to the bone marrow and repopulate the bone marrow. And you will treat the patient that was immune compromised. Okay. All right, a little bit about mesenchymal stem cells. It is a well-characterized population of adult stem cells found in the bone marrow too, as I mentioned. They can form the fat cells, cartilage, bone, tendon, and ligaments, muscle cells, skin cells, and even they're a little more flexible. They can make nerve cells. Okay, so these are the mesenchymal stem cells. Very versatile. You could obtain them in large quantities. That's very important when the quantity is an issue, is a bottleneck. Maintain and propagate it in culture for long periods of time. They can be genetically manipulated. You can incorporate DNA sequences. You can manipulate it. For example, if you take mesenchymal stem cells from a patient and you know what the genetic mutation is on that patient, you can fix that mutation and then put it back into a patient. Okay, so they're very amenable to use and to genetically manipulate. They can be tumorigenic if, for example, let's say you are culturing the mesenchymal stem cells and you have to change the medium, for example, and you say, I can't do it today because I have to do something else. I'll come tomorrow and do it. And you may ruin the experiment because of that. Okay, and so they can develop tumorigenic properties if improperly cultured. And there are clinical trials underway in several clinical centers, etc. So this is what you can do with mesenchymal stem cells. And again, you can separate by adhesion to the plate, cell sorting. Okay, for example, the opposite. You put in these cells here, and you remove the supernatant, and you take the cells that attached to the plate, and you get the stromal stem cells or the mesenchymal stem cells. You have umbilical cord blood stem cells or neonatal stem cells. They're more into the adult stem cells than the embryonic stem cells. They are much more similar to the adult stem cells. They're a rich source of hematopoietic stem cells, less mature than the adult stem cells, but also less mature than the embryonic stem cells. So they're in between. Okay, that's why they are obtained from the umbilical cord. Non-invasive procurement, vast abundance. They can be collected, banked, and they are an alternative source for the treatment of leukemia and other blood disorders. So the umbilical cord blood stem cells are also a very, very good source. They are not as mature as the adult stem cells, but they're very, very usable as well. So adult stem cells in general, you have, for example, neural stem cells. They could be bipotent, tripotent. Okay, they will never be pluripotent. Okay, cardiac stem cells, unipotent or bipotent, not more than that. Okay, or tripotent at the maximum, and very controversial because for example, that if you have a cardiac problem, a cardiac infarct, the heart doesn't regenerate because it's post-mitotic. So where are the stem cells, right? But some people claim that they are inefficient yet, and it's our objective, our ultimate goal, to study the process to see how we can make them more efficient and to regenerate a heart that underwent some cardiac infarct, for example. And then we have the hematopoietic stem cells or the mesenchymal stem cells from the bone marrow. Okay, so this is like the synopsis of the adult stem cells. And now we're going to go back to the pluripotent stem cells and look for alternatives to the many problems that they may have. Nothing is perfect. There are problems with each of them, the embryonic stem cells, the adult stem cells. So one of the problems with embryonic stem cells is the personalized. Can we make personalized embryonic stem cells? Because so far, because of the process, they are not personalized. They should be out of a over the counter. But you don't want over the counter embryonic stem cells. You want patient specific embryonic stem cells. So how do you do that? And people were very smart on this. What they did is they cloned. They, for example, if I want my own embryonic stem cells, what should I do? I will take the nucleus of any cell of my body and put it into an enucleated egg, an egg that in which the nucleus had been removed, right? So I swap the nuclei. My genetic information will go into that oocyte, then will stimulate that oocyte with an electric field to make them go into development. They will start the process of development, and they will make embryonic stem cells. But the genetic information is now mine, right? Because I donated the nucleus there. So I will make my own embryonic stem cells, and then I can use them to treat my liver, for example, if I need more hepatocytes. If I, even further, if I knew a genetic mutation in my pathology, then I could fix that at the embryonic stem cell stage and then put them back into myself, into any organ that, that I need. But the big issue here, the main benefit or advantage is that can make nuclear transfer embryonic stem cells, or we can clone embryonic stem cells. In fact, the process of cloning is successful in monkeys, in Dolly the sheep, for example, in cats, dogs. The president of Argentina, for example, has dogs that have been cloned. Okay, so this is a very, very good thing. But nobody can think of something Frankensteinian, abhorrent, like, for example, cloning humans. Apparently, a technique is there, but don't even try to think about it, okay? That is awful. But this is the process. Originally, it was called therapeutic cloning. Nobody liked that. And now it's called somatic cell nuclear transfer, because there's a nuclear transfer involved, okay? So this is what this slide is about. And this is the schematic. We have an egg where the nucleus had been removed and nucleated, and we have a patient, we have a skin fibroblast, for example. From that patient, a punch biopsy, we take the nucleus and we put it into the egg. And then we stimulate that fertilized egg and we give rise to embryonic stem cells, nuclear transfer embryonic stem cells. We can even fix the mutation if we know it and put it back into a patient. So we close the circle here with the somatic cell nuclear transfer. These are examples. Dolly the sheep here. You have, for example, a surrogate mother. You have the embryo, you have fertilization, you have cell fusion, and you have a donor, a nucleus. And here you have somatic cell nuclear transfer. You have a monkey, macaques, for example, here in China, twins using an oocyte donor here. We enucleated the oocyte here, and we added the fibroblast, not the nucleus, but the fibroblast containing the nucleus, and we activate fusion of the cell membranes, and we obtain a clone. We put this back into a surrogate mother, and then we create a clone. So that is feasible, quite feasible. Okay, this is nuclear transfer embryonic stem cells, or patient-specific embryonic stem cells. Okay, now, what is the problem with embryonic stem cells? There are two problems, two main problems. The first one is that in order to make embryonic stem cell, you have to sacrifice an embryo because of the definition, right? It's an embryonic stem cell. You sacrifice an embryo and you make an embryonic stem cell. So ethically, there's something wrong there. So that's what the one thing, the most important one, right? The second one is the potential to form teratomas, because the embryonic stem cells are pluripotent. They can form teratomas, all right? So I can induce, for example, to make cardiac myocytes, but it'll form cardiomyocytes and bones. And then if I use those for therapy, that's too bad, because I will have calcification of the tissue because of the teratoma potential. So that's not good. So someone, a bright mind, thought about circumventing, bypassing the problem of the teratoma potential and the ethical problems, of the ethical problems, but not the teratoma potential. That is Shinya Yamanaka in Japan. He got the Nobel Prize, and he thought, can I make similar cells to the embryonic stem cells, but not embryonic? In other words, can I force an adult stem cell to become early developmental cell, like pluripotent, which was like a revolution of ideas, because it was he thought of reversing the developmental clock. One can think of any experiment going from egg to birth, but not the opposite, right? He thought that it would be possible to force an adult cell to become a developmental cell. And he succeeded, right? What did he do? He got fibroblasts, and a second experiment from adults, and he added factors, transcription factors, that were important for early stages of development. So he induced a developmental conversion by adding the right type of factors. But which could be those factors? So that's the main question. He started off 20 factors altogether, and he got 2% conversion. He was excited, but 2% is not enough. So he started removing one by one out of the 20 or 14, I don't remember. But he started removing one by one until he got a minimal set of 4, now we call them 4 Yamanaka factors. OCT4, SOX2, KLF4, and CMYC, those factors. You don't have to retain them, the names. But these are the Yamanaka factors. So by doing, and he got the stem cells that were similar to embryonic stem cells, but they were not embryonic stem cells because he didn't use any part of an embryo for this. He didn't sacrifice an embryo. He just converted one adult cell into an embryonic cell. So is there any ethical concern there? Zero ethical concern. And that's why now we are all using iPSLs. We are using ESLs, but we have to report, we have to fill in the blanks everywhere. iPSLs, no concern. You could do whatever you want with the iPSLs. However, they are not identical, they are similar. For example, if you make iPSLs starting out of cardiac myocytes, those iPSLs will have a memory, an epigenetic memory, and would be comfortable going back into a cardiac myocyte stage. They will become sort of pluripotent, but they will have a tendency to go back to the origin. If you make iPSLs from fibroblasts, if you reprogram fibroblasts, they will want to go back to a fibroblast state. So this is an epigenetic memory that is inherent to the iPSLs, okay? And there are other problems. For example, they are tumorigenic as well, in addition to teratomas, since they have c-MYC, they have KLF4, these are tumorigenic factors, all right? So in addition to the potential for teratomas, they have other tumorigenic factors. And it's in the role of us, the scientists, to replace those tumorigenic factors with other factors. Or, for example, nowadays, we use Sendai virus to make the iPSLs. It's a kit. So we get fibroblasts, a kit of Sendai virus, and the cells will become iPSLs. Sendai virus grows as an episomal, as an episome, which means that it doesn't integrate into the genome. So overall, with divisions, the episomes, they do the job, and then they disappear. They're diluted out, okay? So we are perfecting the techniques day by day, and we are making better iPSLs. So that's about iPSLs. These are the ESLs, the gold standard, and these are the iPSLs that are being made out of fibroblasts with the Yamanaka factors and ESL conditions. And then once you have the iPSLs, that they are very similar, but they are not identical to the embryonic stem cells, you have to compare side by side with the gold standard to say, okay, do they do this? Do they do that? Okay, and these are all features of comparison. And sometimes they are a good epigenetic status, but they're not good in teratoma formation. Okay, and then you have to compare the quality of the newly generated iPSLs. This is something that, as I told you before, it hasn't panned out well, but as a concept, is fantastic, and I want to show it to you. Yeah, 10 minutes, and I think we are good in time. So this is transdifferentiation or direct differentiation. So Yamanaka thought, let's make embryonic stem cells like, right? Let's force the system to go from adult to embryonic. There were other investigators who said, if we go back to embryonic, we go back to pluripotency. And we don't want which is good, pluripotency. But we go back to teratoma potential, and that's not good. Teratoma are benign tumors. We don't want them, right? So can we try to force a fibroblast to become, let's say, a cardiac myocyte without the need to go through pluripotency? Can we do that? Can we take that shortcut? It's a huge energetic shortcut, okay? And the answer is yes, it can be done, but with very, very little efficiency. So I think there's a lot of potential in that, because you don't have to go through pluripotency. You can generate from one adult cell, you generate another adult cell. That is very powerful, but yet we are not there, okay? And this is what we have here. So here, for example, Yamanaka's vision was this. I have a fibroblast, I convert it into an embryonic stem cell-like, okay, pluripotent, teratoma, and then I differentiate that iPSL into a macrophage, for example. It could go into a B cell, a beta cell, a muscle cell, etc., etc. We have to develop the right protocol to go down into this energetic sort of mountain. But there were other investigators that wanted to go from a fibroblast to a macrophage directly. And that could be done. And they used a similar technique that Yamanaka used. Yamanaka used pluripotent factors to force the system into pluripotency. These investigators use cardiogenic factors, important for cardiogenesis during development, to force a fibroblast into cardiac myocyte, right? And they had like a battery of factors, and they started removing one by one to get three main factors, cardiogenic factors, to convert the fibroblast into a cardiac myocyte in this shortcut. So that's the concept of transdifferentiation or direct differentiation. Same thing. All right, last one is the synthetic embryo. So you see two embryos here, right? And you're wondering, what does this have to do with the stem cell lecture, right? This is a developmental lecture, but I put it on purpose here because this bottom embryo, this embryo at the bottom, has been made with embryonic stem cells, chest embryonic stem cells. In the right combination, they put, and we learned a lot about this, they used human embryonic stem cells, naive, meaning that untreated, and another embryonic stem cell that had MSX2, which is a factor that is required for the trophectoderm, right? So he combined naive embryonic stem cells with trophectoderm embryonic stem cells, put them into the right cocktail in a bioreactor, and the cells, they reorganized to make themselves an embryo. I think it's 12 days or something like that. So what can we learn with that? Point one, we need trophectoderm for early stages, something that people may ignore. We have ignored the extraembryonic tissues or layers for quite a while. Now we know that if we want to make an embryo, a synthetic embryo, we have to insert the trophectoderm. That's very important. Point two, there's no fertilization, just a mix of cells. There's no sperm, there's no oocyte, there's nothing there, right? That's very important. There's evidence of brain activity and of beating hearts. This is, again, this is a mix of cells here. And And the cells grow in a bioreactor here. They co-aggregate bioreactor and they assemble themselves. And they need the extraembryonic tissues here. There's no fertilization, and they've been named synthetic embryos. And what is the advantage of that? The advantage is that you can't think of manipulating human embryos for these experiments, but it's synthetic embryo. There's no regulation. There's no embryo there. This is an embryo-like. So you could grow this embryo even past potential implantation. This period of development is gastrulation. To achieve gastrulation, you have to go through implantation, right? So using a synthetic embryo, we could achieve gastrulation without implantation. So we can learn a lot about these potentially useful embryos. They can be used to treat infertility, for example, because we can grow past an embryo, past implantation. So there are lots of consequences that can be gathered from these experiences. These are 2-year-old, or they have been described 2 years ago. And that's potentially very, very, very useful, okay? Importantly, no ethical concerns. And I'm gonna leave you here. I thank you very much. Any question that you have now or later when you study, please knock on the door. All right, thank you.